Hi, I'm Sydney,
I'm Alexis.
I'm Viola.
and we have the law enforcement DNA profiling and CODIS topic. So, CODIS is the Combined DNA Index System, which is used by the FBI. It is the FBI database for DNA that comes from all levels, the federal, state, and local. It blends the computer and DNA technology to compare DNA profiles. It utilizes computer software to automatically search for them, and it does not store criminal history, case-related information, or social security numbers or date of birth info. DNA is run through it, and if there's a match found, the lab is notified, and then it is checked again. So then you can see if any cases are linked together. Two types of hits are the offender hit and forensic hit, which is like two or more crime scenes together. There's also cold hits, which gives law enforcement leads in cases that were otherwise thought to be closed or had no other leads. There are three indexes that run through it, which are the convicted offenders, arrestee, and forensics. The amount of samples that CODIS has outnumbered the amount of criminal DNA samples, which can cause backlogs. As of October 2020, more than 200 labs participate. More than 4 million arrestee profiles are in CODIS. There's 12 million offender profiles and 1 million forensic profiles. Matches can occur between offender and or arrestee with the forensic profiles, which would allow enforcement to come up with suspects. Matches between two forensic scenes allow for different jurisdictions to work together. Population profile is a database of anonymous DNA profiles used to determine the statistical significance of a DNA match. The DNA Identification Act of 1994 gave the FBI the authority to establish a national DNA index of law enforcement reasons. CODIS is only in 42 different labs and in only 22 states in the US, which includes Washington, D.C.
So some ethical problems you may run into with this kind of testing is human error in labs. For example, in the Dror and Hampikin experiments, they sent the exact same DNA samples to 17 different experts to see if they would come back with the same conclusion. When the results came back, they were very different results, and so this brought up how labs are not always correct due to the human error. There's also the fear that the wrong people could be dragged into an investigation when they had nothing to do with it due to the human error. There are also privacy issues with familiar searches, so they can impact those wrongfully accused, which will invade on the privacy rights. There's also racial discrimination, where there is more African American men that have their DNA taken and recorded. About 40% of DNA in the databases are actually African American or Hispanic. The DNA database problem also forms ethic biases, or biases being presented in the criminal justice system. So if people from a specific ethnic group have an increase in convicted felons or are arrested or tried, they will be unequally presented in the database. So then this will lead to an increase in arrests with people from that specific background. There are two different kinds of DNA tested. There's the mitochondrial DNA, which is used when looking at the forensic evidence, and there's also genomic DNA, which is the adenine, guanine, thymine, and cytosine. So the different collection methods, so DNA can come from a lot of different body parts, including hair, blood, saliva, teeth, and semen. Law enforcement will use whatever they can find at the scene or get off a person. So in prisons, they tend to take buccal swabs, and then they will send those buccal swabs off to a lab to get tested. There's also tissue samples collected by law enforcement of those who were accused, such as the buccal swabs. A DNA sample is analyzed to compare crime scene evidence, so there's an individual providing a sample for the whole database. So collecting a tissue sample for DNA is mandated only for those convicted of murder or sex crimes, since there is more of an abundance of DNA at murder or rape scenes. There is forced DNA profiling. So under certain circumstances in the US, DNA samples require consent and protection for the donor, so there is no privacy rights being infringed on. There's also DNA database problem that forms the ethical biases that are present in the criminal justice system.
For the DNA identification, most of the human genome is the same, but there are some variations. Variations happen in certain areas that do not need protein coding. Repeated DNA and different lengths appeared in individuals. STR, which is short tandem repeat, contains repeats for short DNA sequences, and it's measured for different individuals. Strength of 13 STR used to identify human remains determines or excludes paternity or matching suspect to a crime. FBI found that people with ethnic backgrounds tend to have an allele of each 13 core STRs. DNA extraction and analysis. When DNA is extracted from a sample, the location that contains the STR is PCR amplified and determined with their size, and this results in the overall profile of alleles. 13 core STRs are different in their strength, analyzes partially degraded DNA samples, and the cost of analysis is reduced because of the amplification of all 13 STRs in 2 multiplex PCR reactions. Different alleles of STR can differ by a single nucleotide. In order to differentiate between single base differences, PCR products can be resolved. Automated DNA sequencing technologies are used to resolve PCR products. This uses software that identifies allele patterns by comparing it to a known ladder. STR match is when 13 core STRs are used for suspect's sample and evidence sample. The suspect would exclude if the samples do not match STR alleles. Then if there's 2 samples that matches the 13 STR alleles, it determines the frequency within the genotype that was observed in the population with calculating the probability, and you would first calculate the frequency with each STR allele, and that happens in individual ethnic groups.
Then here are some fun facts that we have found about DNA profiling in law enforcement in the US and throughout the world. PCR gel is read at the end of the processing sequences after everything is completed. It separates out DNA fragments and it looks like ladder rungs, which are called bands. The bands are compared to other known samples to see if there's a match or not. It costs about $500 for the collection, getting it from the person and everybody involved in the sequencing of it. And then through the entire process, it's about $2,000. CODIS gave the Malaysian government a model to establish their own forensic testing. And then DNA profiling is used throughout the world. However, there are not as many national databases consisting of both crime scene profiling indexes and convicted offender DNA profiling indexes in developing countries. Developing countries do not have as wide of a DNA database. This kind of testing can focus investigations and can lead to shortened trials and lead to guilty pleas. DNA profiling is not an entire person's genome that is being read because it would take too long to process the entire genome. It is based on a statistical probability. If a DNA is not lawfully or ethically collected and tested during court cases, the DNA can be refused as evidence, which has been seen in a Supreme Court case of Minnesota in 1989.